Let's talk glycolysis. Glycolysis is the first step in a process known as cellular respiration. And cellular respiration, here's the overall reaction, is a catabolic process in which we get energy into a form that we can use, known as ATP. So, in this, we start out with a carbon source, which is going to be sugar, and we're going to combine it with oxygen, which is our electron acceptor, and we're going to get out CO2, we're going to get out water, and we are going to get out energy in a form we can use, ATP. This breaks down into three main processes: glycolysis, the citric acid cycle, otherwise known as the Krebs cycle, and oxidative phosphorylation, otherwise known as the electron transport chain. The second two steps don't happen in every organism. It depends on who you are if you do those two steps. But everybody, absolutely everybody, from us all the way down to bacteria, everybody does glycolysis. So let's take a look. Glycolysis, glyco, sugar, lysis, breakdown. So we are going to break down sugar into something we can input into the citric acid cycle. So let's start looking at it. We are going to start with an investment phase, and the investment phase means that we are going to put in ATP, or put in energy, and then later on we're going to get out more of it in the payoff phase. So we're going to start by phosphorylating glucose. And this is important because when glucose comes into your cells, it comes in through the GLUT transporters, and it comes in by itself, right? So why doesn't glucose just diffuse out of the cell again? Well, when it gets phosphorylated, all of a sudden these transporters can't recognize it and carry it anymore. So the phosphorylation, in addition to activating it and giving it energy, it is also a protection against the glucose just floating on out of your cells again. So we're going to use hexokinase in order to make glucose 6-phosphate. And then we are going to do a bit of isomerization. We're going to turn glucose 6-phosphate into a different sugar known as fructose 6-phosphate. So this has all the same chemical formula, just a little bit of a different arrangement. Then we are going to continue with our investment. We are going to put in another ATP, so we're going to phosphorylate this again using phosphofructokinase, and we are going to make it into fructose 1,6-bisphosphate. And here comes a really important part, because we are going to take fructose 1,6-bisphosphate, and using our aldolase, we are going to split it up into two things. So from now on, all the steps we do are multiplied by 2. So we are going to make two things. We're going to make G3P, glyceraldehyde 3-phosphate, and we are going to make DHAP, dihydroxyacetone phosphate. And DHAP can actually be isomerized into G3P. So we're going to end up with 2 G3Ps, and these 2 G3Ps can go on into our next phase, which is the payoff phase. So at the beginning of the payoff phase, we're going to do something that's important to the rest of the cellular respiration process. So if you go beyond glycolysis and you do the citric acid cycle and you do the electron transport chain, you are going to need electrons. And these electrons are shuttled over to the electron transport chain via NADH and FADH2. In glycolysis, we're going to start by using NADH. So we're going to have NAD+, and we're going to take 1 hydrogen and 1 electron pair, and we are going to put them in as NADH. And these are going to get shuttled later on into the mitochondria and into the powerhouse of the cell to do their thing and make ATP. So we're going to go on, and we're going to remove some phosphates, and we're going to get ATP out of it. So here is the payoff, and this step kind of shows ADP into ATP with phosphoglyceralkinase. But what's actually happening is we have 2 ADPs turning into 2 ATPs, right? Because from now on, ever since we made our DHAP and we made our G3P, we're multiplying everything by 2. So we're going to do another step with phosphoglyceromutase. We're going to make 2-phosphoglycerate, and then we are going to release some water. So our enolase is going to release water and do a little bit of isomerization, or actually tautomerization. And our next step, we are going to get our last 2 ATPs out of this. So in our net total, we lost 2 ATPs, but we gained 4 ATPs in making our final product of pyruvate. So we're getting 2 pyruvates. So here's a little tally of everything we got. We made glucose into 2 pyruvates, we released some water, we made 2 ATPs net, and we made 2 NADHs. So these numbers are probably going to come up if you're in a more advanced biology course. This is going to come up and you're going to want to know the numbers for this and for cell respiration in general. So if we look at the regulation of this process, glycolysis is regulated in a few different ways. The first way, and I want to make a whole video on insulin and glucagon and that whole regulation process, but to put it simply, insulin is like a key that opens the doors for glucose to enter the cells. So I talked briefly about the GLUT transporters, and the GLUT4 transporter can actually be activated to let in glucose when there is high insulin, and high insulin happens when you have a high blood glucose concentration. So this is going to trigger more glycolysis because you have more glucose in the cell. So if you're looking at this, we have glucose, and we go through glycolysis to make pyruvate. And not everything is reversible in this, but we do have a different process called gluconeogenesis that makes pyruvate back into glucose. So this is essentially an equilibrium. So if we put more glucose into our cells, this is Le Chatelier's principle, right? This is saying that if we add more glucose, the equilibrium is going to want to shift toward the products and do more glycolysis. So that's the way the insulin ups our glycolysis rate. ATP is also going to play a role. Think about it, this whole process, glycolysis by itself and the whole cell respiration thing is making ATP. And usually in biology, whenever we make something, that something is going to go back and inhibit the process so we don't exhaust it all out and make way too much. I mean, you can't have too much ATP, but you can be wasteful by doing a process too many times. So this is called feedback inhibition, and ATP is going to actually inhibit the activity of phosphofructokinase, which is one of the first enzymes we saw that phosphorylated fructose 6-phosphate into fructose 1,6-bisphosphate. And this is going to lower the activity of glycolysis because if we have one enzymatic process that's stuck, then things are going to be backed up down the line as well. Another thing that's going to inhibit this same enzyme is fructose 2,6-bisphosphate. Now remember that what we actually see in glycolysis is fructose 1,6-bisphosphate. So this is a slightly different kind of thing. But it is going to inhibit, or not inhibit, it's going to activate this enzyme because this is similar to the metabolite fructose 1,6-bisphosphate that comes before the enzyme. So this is the opposite of feedback inhibition. This is a reactant or something that is chemically similar to the reactant pushing everything forward. So here are two graphs that kind of show what we were just talking about. So we've got fructose 2,6-bisphosphate activating this enzyme and ATP inhibiting this enzyme. So take a look at those, screenshot them if you have to. This is an important mechanism of glycolysis regulation. So now I forgot to include a diagram that I wanted to include where we have glycolysis, right? We're going to take a minute to look at the big picture. So in glycolysis, we get out some NADH. Pyruvate's going to be made into something called acetyl-CoA. Don't worry about that yet. And acetyl-CoA is going to go through the citric acid cycle. And it's going to output some stuff. It's going to output CO2. And we're going to get some more NADH, FADH2. And these guys are all reduced forms. So you have NAD plus and you have FAD, and when you put these hydrogens along with their electron pairs on there, you are reducing them because remember, oil rig oxidation is losing, reduction is gaining. We are gaining electrons. So all of these fine little, I like to call them shuttle buses. That's how I learned this. They are shuttling all these electrons and their protons They're all going to go into the mitochondrion, and the mitochondrion, I mean, actually, no, this process happens in the mitochondrion, as well. So this whole thing is the mitochondrion. But anyways, this is where oxidative phosphorylation is going to happen. And all of these electrons are going to go into the electron transport chain, where they're going to be used to power the ATP process. And in doing so, they are going to output NAD plus and FAD. Because now our hydrogens are used up in the proton pump. We're going to learn about that later. The electrons are used up in the electron transport chain. We've made our ATP. We're happy. And the important part is that now these electron carriers or shuttle buses or whatever you feel like calling them can go back and be recycled in order to do everything over again. So why does this matter? Well, sometimes in anaerobic organisms, which are the ones that don't use oxygen, or in aerobic organisms like us, when we are hypoxic, when we don't have a lot of oxygen, we can't do this whole oxidative phosphorylation thing. So we get backed up, and the only thing we can really do is glycolysis. So here in electron transport chain and oxidative phosphorylation, we get big ATP. We get a lot. This is our main source. In glycolysis, we get little ATP. We get net 2 of them, right? Whereas the whole thing, we get 32 to 34 in the total cell respiration process. But when we don't have oxygen, we need to get ATP from somewhere. So we are going to get it from glycolysis. Except if we can only do glycolysis, we're using all those NAD plus and we're getting out all these NADH, but the NADH, they're not giving their electrons away, right? They're not giving them to cell respiration, the electron transport chain. So we are stuck with a bunch of NADHs, and we don't have any NAD plus to make glycolysis work. So we have to find a process that can get us back our NAD plus from NADH without the whole powerhouse of the cell, so that we can go do glycolysis over again and get little ATP enough times that it works for our cells. So how do we do this? We do this in a process known as fermentation. So the whole objective of fermentation, like I just said, is to get our NAD plus back so that we have reactants for glycolysis. And there's 2 different ways of doing this. For us, what we want to do is lactic acid fermentation. So we are going to take our pyruvate, so here's pyruvate, and we are going to make it into lactate. And in doing so, we have this other process that gets us back our NAD+, and we are able to do glycolysis again and get a little bit of ATP, but it's better than nothing, right? In some other organisms, such as yeast, or I don't know if it's bacteria or what, but it's not complex eukaryotes like us, they do ethanol fermentation, so they have a little bit more of they have more steps in their process. They've got this little intermediate here. So they'll take pyruvate, they're going to make it into acetaldehyde, and acetaldehyde is going to get made into ethanol. And if you look at pyruvate, pyruvate's a 3-carbon thing. Acetaldehyde, acet means 2. So we are also going to get CO2 out of this. So these are the 2 fermentation processes. And this lactic acid, a question you might see come up in a biology test is like, why do our muscles hurt when we exercise? The answer is the buildup of lactic acid that happens when our muscles run out of adequate oxygen and have to start doing this fermentation process. So that's glycolysis. I hope this helped. I'm going to try to put out videos of citric acid cycle and electron transport chain soon, so watch those as well.